Hello everyone, welcome back to my YouTube channel. Today we are going to see Maharashtra State Board Class 10 Science 2 Chapter number 8 Cell Biology and Biotechnology and its applications. In the first part already we have discussed about the stem cell and the various uses of the stem cell. Today we are going to talk about the biotechnology and the various terms and the things related with the biotechnology. As in earlier classes already we have talked about the biotechnology. It's nothing but bringing about the artificial genetic changes and hybridization in organisms for human welfare. In a simple words, the changes which can be possible in the genes with the help of the technology is called as the, or that entire process is called as the biotechnology. In this particular section, we are going to talk about the biotechnology, which the included main areas under this particular section. In that, the first what we have that is the uses of a various abilities of microbes like a yogurt production from milk and alcohol from molasses. Use of productivity of the cells, for example, production of the antibiotics and the vaccines, etc., with the help of these specific cells. Use of biomolecules like DNA and proteins in human welfare. Development of the plants, animals, and products of a desired quality by gene manipulation. Production of a human growth hormone with the help of the genetically modified bacteria. Use of a genetic and non-genetic technique. Non-genetic biotechnology involves the use of either cell or tissue. For example, tissue culture, production of a hybrid seeds, etc. So these are the main areas where the biotechnology helps a lot. Further, next what they have given is about the benefit of biotechnology. What is a benefit actually? That is a, we can use it, that is different thing. But what is the benefit of that they have given over here? It has become possible to increase the hectare yield irrespective of the limitations of cropland area. Further is that is expenses on a disease control have minimized since development of resistant varieties. Due to development of fast fruit setting varieties, yield per annum has been increased. Development of a stress resistant varieties which can withstand variable temperature, water stress, changing fertility of soil, etc. has become possible. Means the external factors which affects the agriculture that can be controlled or that can be tolerated by the plants. Further, they have given us about the development of a biotechnology in India. The government of India has established a National Biotechnology Board in 1982. National Biotechnology Board in 1982. This board was transformed into a Department of Biotechnology under the Ministry of the Science and Technology in 1986. So within 4 years, they changed the complete concept of it and they made it as a Department of Biotechnology. Further is it includes the National Institute of Immunology, National Facility for Animal Tissue and Cell Culture, National Center for Cell Science, and the National Brain Research Center, Central Institute of Medicinal and the Aromatic Plants. These are the facility of a higher education and research in these institutes from where thousands of students have pursued PhD degrees and are contributing to the progress of country in the field of biotechnology. Now, after that, the commercial applications of biotechnology, the commercial uses. In that, the first what we have that is the crop biotechnology, which is used in agriculture field to improve the yield and variety, hybrid seeds to give the better crops. Genetically modified crops means the well developed seeds are used to produce the good quality of the products. Then after that, the BT cotton, they have taken the example over here. A gene had been isolated from the bacterium bacillus thuringiasis and the integrated with the gene of a cotton. Due to this, the toxin which is fatal for bollworm was produced in leaves and bolls of cotton. If bollworm worm feeds on leaves, the toxin destroys the alimentary canal and the bollworm dies. Further, B2 brinjal varieties is developed by using the gene isolated from Bacillus thuringiensis. This improved variety of a brinjals kills the pest in same way as the BT cotton does. Same way about the rice also, a gene synthesizing the vitamin A, that is beta carotene, has been introduced in the variety of a rice as compared to the normal variety. The variety which has been developed in 2005 contains 23 times more amount of a beta carotene. Herbicide tolerant plants. Weeds always affect the growth of a main crop. If herbicides are used to destroy the weeds, it affects the main crop too. Due to these, herbicides tolerant plant varieties of crops are being developed. Due to this, it has become possible to selectively destroy the weeds. Further, they have given us about the biofertilizers, one of the most important part of the agriculture now. Due to use of a biofertilizers instead of a chemical fertilizers, nitrogen fixation and the phosphate solubilization abilities of the plants are improved. Mainly the bacteria like Rhizobium, Azotobacter nostoc, Anobina, and the plants like Azolla are used as biofertilizers. In the last year, we have studied about the tissue culture. Genetic improvement of the plants has become possible due to the tissue culture, and besides those characters inherited to the next generation. In the ninth standard already we have studied about the inheritance of the traits. Further is that is the animal husbandry. They have given two main methods are artificial insemination and the embryo transfer are used in animal husbandry to improve the both quantity and the quality of the products from the animals. Further, that is the human health. Diagnosis and a treatment of the diseases are two important aspects of the human health. In that, biotechnology helps to identify the role of a gene, and then the various diseases can be controlled or can be treated with the help of this particular technology, which we call as the biotechnology. Examples are like a diagnosis of a disease like a AIDS, dengue can be done within few minutes. Further, that is the vaccines and vaccinations. Vaccine is the antigen containing material given to the acquire either permanent or temporary immunity against a specific pathogen or disease. Traditionally, vaccines were prepared with the help of the pathogens completely or partially. Killed pathogens were used as vaccines. However, due to this, there were chances of contracting the diseases in case of some persons. Hence, an alternative that is scientists try to artificially produce vaccines with the help of a biotechnology. For this purpose, scientists produce the antigen in laboratory with the help of a gene isolated from the pathogen and used it as vaccine. The safer vaccines are being produced. Further, that is edible vaccines also. Work on production of edible vaccines is in progress, and presently potatoes are being produced with the help of biotechnology. These potatoes are called as the transgenic potatoes. These potatoes will act against bacteria like Vibrio cholera, Escherichia coli. Consumption of these raw potatoes generates immunity against cholera and the disease caused due to E. coli. And further, they have given us about how these potatoes are produced. Actually, isolation of a desired gene from the human pathogen, then the transgenic plant virus infects the pieces of the potato leaves. Entire plant is developed from the leaf pieces containing gene of human pathogen, and then the consumption of raw potatoes helps to develop immunity against that pathogen. Further, that is a treatment. Biotechnology is useful for the production of hormones like insulin, somatotropin, and blood clotting factors. Interferon, this is group of a small sized proteins molecules used in a treatment of viral diseases. Gene therapy, gene therapy to treat genetic disorder in somatic cells has become possible due to technology. For examples, PKT, that is a phenylketonuria, arises due to genetic changes in the hepatocytes, that is the liver cells. So, further, that is the cloning, production of the replica of any cell or organ or entire organism is called as cloning. Reproductive cloning, a clone can be produced by fusion of a nucleus of somatic cells with the enucleated ovum of antibody. Thus, there is no need of a sperm to produce the new organism. Next, that is a therapeutic cloning. Stem cells can be derived from the cell formed in laboratory by the union of a somatic cells nucleus with the enucleated egg cells. Various diseases can be treated with the help of these stem cells. Proteins produced by the biotechnology and the names of diseases they are used against insulin for the diabetes, somatostatin for the dwarfism, erythropoietin that is anemia, factor 8 for that is for the hemophilia, interleukin that is for the cancer, and the interferon for the viral infections. Further, they have given us about the industrial products that is white biotechnology. Various industrial chemicals can be produced through less expensive processes. Example, alcohol production from a sugar molasses with the help of the transgenic yeast. And even the environment and biotechnology, that relation also they have given. But before that, one information they have given over here is about the a sheep dolly was born in Scotland by cloning technique. The first animal which got the birth by the cloning system, that was the dolly, the sheep in the Scotland. And this happened on the 5th July 1996. Nucleus from the udder cell of sheep of a Findorest variety had been introduced into enucleated ovum of a Scottish sheep. Then the ovum was allowed to develop in the uterus of a Scottish sheep, and thereby the dolly had been born. It was showing the characters as per the chromosomes in nucleus, and any character of a Scottish sheep was not visible. Further, that is the environment and biotechnology. It has become possible to solve environment related various problems with the help of a biotechnology. In that, the microbial technique are already in use for treatment on sewage and solid waste. Sewage is rich in organic matter. If such sewage is released in natural water bodies like rivers, organic matter, it is in its gets oxidized with the help of a dissolved oxygen. Due to this, level of a dissolved oxygen in the water decreases, and that will affect the organisms which are living in that water. So the remedy of this is what the oxidation of the water should be avoided, and that's possible with the help of the microorganisms, with the help of a biotechnology that can be treated and controlled. Further is bioremediation, in that they have given all this information, that is pseudomonas, then the teres, genetically modified variety of a human mustard can absorb selenium from soil, then the sunflower can absorb uranium and arsenic, then the bacterium, that is a Deinococcus radiodurans, is highly radiation resistant organism. It has been genetically modified and used to absorb the radiations. Further, they have given us about the food biotechnology. Food items like a bread, cheese, wine, beer, yogurt, vinegar are produced with the help of microorganisms. DNA fingerprinting. DNA sequence of each person is unique as that of the fingerprints. Due to this, identity of any person can be established with the help of its available DNA. This is called as DNA fingerprinting. Further, they have given us about the cleaning of oil spillage in oceans. This is one of the most important use of the biotechnology. If oil spillage occurs, it adversely affects the marine life. Now cleaning the ocean without any harm to environment in cheaper way has become possible with the help of the oil digesting and fast multiplying bacteria. India born American citizen and scientist Dr. Anand Mohan Chakravarti had, for the first time, suggested the use of such microbes. Naturally, the credit for this discovery goes to him. So, this is about the use of the biotechnology and biotechnology itself. In the next video, we are going to talk about the important stages in the agriculture development.